In this question, we have been given some Escherichia coli genes involved in DNA replication under column X and their functional eukaryotic orthologs under column Y. We have to match the correct combinations. Let's look at each of the options one by one. Functionally, DNA B is a bacterial helicase, which is an enzyme that unwinds the double-stranded DNA at the replication fork, enabling the separation of the two strands so that they can be copied. Similarly, the mimic chromosome maintenance complex or the MCM complex acts as a DNA helicase in eukaryotes. Structurally, DNA B is a ring-shaped hexamer composed of 6 identical subunits. In eukaryotes, the MCM complex is a heterohexameric ring composed of MCM2 to 7 proteins. If we look at the mechanism of action, both DNA B and the MCM complex use the energy from ATP hydrolysis to unwind the DNA helix. Therefore, the functional ortholog of DNA B in eukaryotes is the MCM complex. Now let's take a look at DNA C. In E. coli, DNA C is a crucial protein that loads the DNA B helicase onto DNA to initiate replication. In eukaryotes, the function of DNA C is carried out by a combination of proteins called CDC6, CDC18, and CDT1. Specifically, CDC6, like DNA C, is a member of the AAA plus family of ATPases. CDC6 plays a similar role to DNA C in recruiting the MCM2 to 7 helicase complex to replication origins. Therefore, the functional ortholog of DNA C in eukaryotes is CDC6. Next, let's discuss the beta clamp. In E. coli, the beta clamp is a dimeric ring-shaped protein that encircles the DNA and facilitates processive DNA replication by tethering the DNA polymerase to the DNA. In eukaryotes, the functional ortholog of the beta clamp is proliferating cell nuclear antigen or pCNA. pCNA is a trimeric protein that also forms a ring-like structure similar to the prokaryotic beta clamp. It acts as a sliding clamp that tethers DNA polymerases to the DNA template, increasing processivity. Moreover, both the E. coli beta clamp and eukaryotic pCNA are involved in more than just DNA replication. They also participate in DNA repair in other cellular processes. Let's move on to DNA G. DNA G is a prokaryotic primase that synthesizes short RNA primers on the leading and lagging strands of DNA. These primers provide a 3' OH group for DNA polymerase to initiate DNA synthesis. In eukaryotes, the functional ortholog of DNA G is DNA polymerase alpha or Pol alpha, which also acts as a primase. It synthesizes RNA primers on both leading and lagging strands, allowing DNA polymerases delta and epsilon to extend these primers and complete DNA synthesis. So, the functional orthologs are DNA B and MCM complex, DNA C and CDC6, beta clamp and pCNA, and DNA G and Pol alpha or primase. So the correct combination is the third option. Do visit my YouTube channel, Simply Biology with Manisha, for more such videos. Thank you.